There are two types of people when it comes to silicon. There are those that don't know, and there are those that don't know that we know. Okay? And so that's what we're trying to do, is bring some of the people that know so we can educate those that don't know. I'm going to carry on and talk about silicon and biotic stress and focus primarily on suppressing plant diseases because I'm a plant pathologist. We'll start with Insuki Odenera. This gentleman in 1917 in Japan decided he wanted to know what the chemical analysis of a rice plant was. So he went out to about 13 provinces and collected plants. And he noticed as he was collecting them, some were diseased, some were not diseased. And when he started to do the analysis, he found out the ones that had less disease from rice blast were high in silicon. So that was probably, in 1917, was probably the first association of this element with a plant disease. Magnaporthygresia, there's two phases of it. There's a leaf phase, we call it leaf blast, and then there's a panicle blast. And you'll be seeing this throughout the presentation. I come along in the 1940s, Wagner. I'm not sure if he was reading the Japanese scientist's papers, but he started to look at this, and he was able to show that they were using sodium silicate and calcium silicate, that he was able to reduce the number of conidial colonies and necrotic spots caused by powdery mildew. And so that was probably the first evidence in a dicot. Since that time, rice has been the model crop for monocots, and cucumbers have kind of been the model crop for dicots when it comes to studying a number of plant diseases. But since that time, there's another important agronomic and horticultural crops that have been looked at, and a number of the diseases that have been infecting those have been investigated. Some of these plant pathogens you can see are predominantly by plant pathogenic fungi, but there are some that are from the Oomycota that are fungal-like, like Phytophthora pythium, bacteria like Ralstonia, xanthomonas, viruses, and nematodes. I'll point out a couple of these. Let's say meloidigine. This is coming out of Brazil on coffee, and they've been able to show some reductions in gall numbers as well as in egg numbers, which is very important. Similarly, Scott Leisner and Jonathan Krantz, along with Wendy Zellner, they've been able to show that tobacco ring spot virus, been able to show not only a delay in the onset when the disease first appears, but also there's a timeline in suppression. Ralstonia is coming out of Germany, showing differences in cultivar susceptibility with or without silicon. And as you see, as the more resistant the cultivar is, silicon seems to enhance that resistance. Phytophthora, this is on peppers, and you can look in the middle where you see plus silicon versus minus silicon, and this is a crown and root rot pathogen, and you can see that the progression up the stem is much less, and the impact of the pathogen on the roots is much less in comparison to the non-amended control. And similarly with Colletotricum and Anthracnose on cucumbers, get reduction. And I'll just take one more fusarium, another root pathogen in tomatoes, most recently showing with and without silicon, the difference in the cortical root rot and lateral root rot. So it seems as if you go to a soil like histosoils that we did research on in South Florida, as you increase rates of silicon in the soil, you will get a curvilinear response where it reaches asymptote at the 10 metric ton rate of silicon in the plant tissue. So you're going from less than 1% in the controls to like 6% where it's been amended. And similarly, as you increase the rates, you'll get either a linear or a curvilinear decrease in rice blast incidence. And interestingly, this line represents a one-year residual. So a one-year residual, you're getting a linear response, and we have a fresh application, a curvilinear response. We've seen similar examples in turf. As you increase concentrations of silicon in the soil, you get a nice linear increase in the concentration of silicon in the plant tissue. And if you put that relationship to disease development, again, as you're increasing the concentration of silicon in leaf tissue, you get a nice linear decrease in the amount of disease that's occurring. So it seems like it's probably concentration dependent, and this is a very nice study that kind of shows that. This was done with Fabricio's group in Brazil. This is the LSI1 mutant, which means that it's deficient in its ability to accumulate silicon versus the wild type. So in this case, where you did amend with silicon, you got about 179% increase in accumulation versus the wild type, where it was about a 220% increase. But the difference between the wild type and the mutant was about 112%. You can see the control and then amended on the wild type, and then the control and the amended on the mutant. For example, even though you got a significant difference in the amount of disease that was occurring, it was only about a 12% decrease. But where you had in the wild type, it was like a 35% decrease. So concentration matters. And the difference even between these was about 23%. So concentration is very important. And in this case, the concentration how it's measured is the insoluble silicon on a dry matter basis in the leaf tissue. Recently, Leisner and Franz and Wendy, they showed that this is really interesting. Here's a pathogen, a virus infecting plants, and it seems to have the plant accumulate even more silicon in the presence of this virus. So there's situations like that we're starting to understand a little better about what's going on in this element and how it's affecting certain plant diseases. As this kind of went along, Bellinger's group in Canada, they felt like the supply of silicon had to be continuous or wasn't very effective. So for an example, they had a treatment where there was no silicon amended, another one where it was silicon amended, and then they switched it. There was none, and then it was amended, so you got no disease, as you would expect. But then when you converted that solution from with silicon, then without, you got disease. And since it doesn't really polymerize in dicots like it does in monocots, it made you think that maybe it's the soluble content of the silicon that's important in disease suppression. And I'll come back to this more later. There's a similar study, again, with this continuous silica supply, and had to do with Pythium phantomatum infecting bitter gore. In this study, of course, they had their controls without silicon, with silicon, and then with Pythium, and then silicon, again, challenged by Pythium. And in the subapical region of the root, where silicon is high in the symplast, again, this is the control, and this is where silicon has been amended. Both are challenged by the pathogen. You get a reduction in the amount of infection that's occurring. This is, again, with a continuous silicon supply. And similarly, in the bicipital region of the root, the same thing was occurring. But if you started this silicon supply right at the same time that you inoculated the plant, again, in the subapical region, there was no differences between silicon amended, non-amended, challenged by the pathogen, as well as in the bicipital region of the root system. So we were curious, again, approaching this soluble, insoluble aspects of silicon and how it's affecting plant diseases. So we set up a study similar to what was done with the Bellinger's group in cucumbers with rice. And what we did was, around 24 hours before inoculation, we changed our plus silicon to a minus silicon, and our minus silicon to a plus silicon, with the idea that now this would be the insoluble form of silicon, and this would be the soluble form of silicon. And 24 hours after we changed the nutrient solution like that, we went in and we measured it, and you can see where we had continuous silicon supply. It was just like the supply when we switched out the minus silicon to plus silicon. And then we were able to make the plus, which was plus silicon, now minus. The xylem concentration in silicon sap was just like the control. And similarly, but maybe not quite as clear, you did get a reduction in the amount of insoluble silicon where we switched it out. And as we would expect, you wouldn't expect where we added silicon that the insoluble would still be lower, but not quite like the control. But again, we're showing the reduction. And so when you look at the progress of leaf blast development, this is the control. This is the minus silicon plus silicon. This is the plus silicon minus silicon, and the plus plus silicon. And what we can conclude from this is that both the soluble and insoluble forms of silicon are very important in controlling rice blast development. But if it's continuous, probably both are functioning in this case. So what about silicon and how it affects host plant resistance? This is a study that was done with sheath blight. We had 2 cultivars. These are considered to be susceptible. These 2 cultivars are considered to be moderately susceptible, and these 2 have high levels of partial resistance. Our red is the control, and it kind of follows that. Susceptible, moderately susceptible, partially resistant. When you amend with silicon, you got reduction across all the cultivars and disease development. But what's interesting to me was you were able to take the susceptible cultivars and the cultivars that were moderately susceptible and push that level of resistance to the same level of high partial resistance. So this has implications for managing resistance under field conditions. Similarly, we did this with genotypes that were susceptible, partially resistant, and completely resistant to rice blast. We amended the soil with different levels of calcium silicate. And what we found out was at the highest rate, we could take the susceptible cultivar or Cuticle-1 and push its level of resistance to the same level of resistance as Linear 2. And then with Linear 2, at either one of these concentrations, we could increase the resistance to the same level as almost complete resistance. So again, this has implications for managing cultivars under field conditions, because in plant breeding, it takes a long time to put certain traits into a plant, yield, amylose content, and you may have resistance to one pathogen, and then when you put that out in the field, there are always others that can appear. And so this could play a very important role. When you say resistance in the world of plant pathology and breeding, we talk about components of host plant resistance. And this is made up of the incubation period, from the time you see symptoms to the latent period when you start to see sporulation, lesion lengths, lesion expansion rates, the number of sporulating lesions per millimeter squared, and the number of spores per millimeter squared. And in the case of rice blast, silicon affected all these components of host plant resistance, which suggests that it's having an effect on the plant. So there's got to be some type of structural or biochemical or genetic role for this element. And this kind of shows that deposition of silicon just below the cell wall. Here's the control. One of the theories behind the resistance with silicon against plant diseases was the mechanical barrier hypothesis. And the idea was you'd have a conidium land on a leaf surface, it'd form a germ tube, and then an appressorium, but there would be no infection. So the idea was that this deposition of the silicon layer below the cuticle is uniformly distributed. And there has been some papers that were just published in 2008 still supporting this theory, where they showed the relationship of, as you increase silica gel to the plant, you saw a decrease in the proportion of appressoria, the rice blast pathogen in the motor cells. So there is still some evidence for this mechanical barrier hypothesis. However, we did some work a number of years ago. This is 96 hours after infection. You can see how large these lesions are versus where it's been amended with silicon and they're like pinpoint. And if you go under transmission electron microscopy, you can see the fungal hyphae. They look pretty normal. They're colonizing. You see a little degradation of the cell wall. But where it's been amended with silicon, the fungal cell, it almost looks like a vacuole. It's totally devoid of any cytoplasmic material. And it's surrounded by this amorphous material that when you stain it with toluene blue indicates it's a phenolic. And phenols are one class of compounds that are known to protect plants from pathogens. Similarly, we did some work with HPL analysis where we extracted subfraction from plants that have been amended and non-amended with silicon and challenged by the pathogen. And basically, so here's the pathogen, here's with silicon, and you can see this zone of inhibition. And right here is where 50 microliters of that subfraction was placed. And 46 and 47 minutes after elutriation, mammalactone A and mammalactone B were found. And these are phytolexin compounds, and phytolexins are known to have antimicrobial activity against this pathogen. In fact, there's about 4 or 5 other phytolexin compounds that have also been identified in rice. We went further and did some work with RNA, with analysis of the transcript accumulation for defense-related genes using northern blot. M21 is a highly susceptible cultivar. It has no known major or minor genes for resistance. So there's silicon. It was on a time course study, and at 36 hours, it was very strongly constitutively expressed for beta-1,3 glucanase. And you can see it's about a 5-fold increase in the control versus where it was amended. And glucanase is very important because many fungi have glucan in their cell wall. So there's another defensive compound produced. Similarly, again, we have our M201, it's susceptible. There's our control with silicon. And it's around 60 hours that we started to see that was more strongly constitutively expressed in comparison to the control for peroxidase. And peroxidase is very important in lignin production in plants, protecting the plant from pathogen infection. And again, there were large, this is the relative intensity, and it's about a 5 to 10-fold increase in the intensity. And looking at PR proteins, these are known to have antimicrobial activity. These were detected at 60 hours, and they were much more strongly expressed in comparison to the control, maybe double. So based on this, there's probably some kind of molecular basis for silicon-mediated disease resistance. So silicon, does it change the rice gene expression? Does rice change the way it reacts to a pathogen infection? So we did this study where we made these comparisons with silicon pathogen, a control, and then just a pathogen. And basically, 24 hours, messenger RNA was extracted, it was labeled, broken into 2 groups, and then hybridized to a 44K gene chip, rice chip. What we have here, this is called a volcano plot. So what this shows is the distribution of the data, the levels of expression, as well as the number of differential genes that were up and down regulated. So in this volcano plot, anything below this line is not significant. Up here, this is less than a 3-fold change, and anything to the right or left is a greater than a 3-fold change. And on the right, these are genes that are upregulated, and on the left, genes that are downregulated. So this is for the control versus silicon. So you can see right away, even without a stress, there's a number of genes that are up and down regulated. Here's the comparison showing pathogen versus the control, and then here's silicon versus silicon pathogen. And you can see a big difference in the number of data points that are being expressed. So you can say that silicon does appear to be having an effect on the expression of the rice plant. There were 440 fewer genes expressed in the silicon versus silicon pathogen comparison in comparison to the control versus the pathogen. So there was an effect on the host and pathogen, and they're also a unique set of differentially expressed genes. Here's the distribution of some of these expressed genes. So here's our control versus the pathogen. There were over 700 expressed genes, but there were 312 that were uniquely differentially expressed, and this is a comparison into the other comparisons. And similarly, even though it says like 54 for this combination and 54 for this combination, these genes are uniquely differentially expressed from each other. So it shows these are pathogenicity and stress genes that were here, they're upregulated, this is like downregulated, and these are some that are unknown and then other. So what were some of those? Well, for silicon versus control, we saw a salt-tolerant-like protein. With a silicon pathogen versus silicon, we saw peroxidase. I just showed that to you earlier with the northern blot analysis. Peroxidase, the antifungal protein, we saw that with the PR1 proteins, for example. Of course, this is not unique to rice. Some of the other studies that have been done is in the tomato bacterial wilt, wheat powdery mildew, and then Arabidopsis and powdery mildew. And there are a number of uniquely expressed genes, whether they're defense or stress related, that have been observed. So if you look at rice, you may say, well, how does it increase rice resistance to blast? Well, if silicon is deposited below the cuticle uniformly, then it's probably difficult for that pathogen to penetrate. But if you look at a number of X-ray microanalysis, you can see that it's not uniformly distributed. And so it does penetrate in areas where there may not be silicon below the cuticle, but there's still that defense response. And so it's probably both could be functioning in rice. So what about managing this under field conditions? So we were curious about silicon and fungicides. We had a study where we looked at silicon alone plus a benlate. That was back in the late 80s, early 90s. That was the fungicide of choice and the only choice. And you could see that it was performing as well as a fungicide and suppressing panicle blight. But again, the combination was the most effective. This was a study in the upland in Ciot, Colombia. So this is the control, fungicide, and then silicon. And you can see that we had a very heavy blast infection, and that's why the rows haven't closed in. Those leaves are infected and withering and dying. And we did it at different rates. But the bottom line is, again, that a full rate of fungicide and silicon alone was as effective. So it may mean that you can maybe manage your fungicides. You don't need to apply it as often, or maybe in a leaf blast phase you don't need to use it. We did a further study where we looked at a 1-year residual, and we compared that to a fresh application and then full rates of fungicide. So here's a 1-year residual. And so what if you pay $100 in year 1, but it's effective in year 2? Well, then it amortizes. It costs you $50. So what are some facts about silicon and biotic stress? Silicon concentration, whether it's insoluble or soluble, as it increases in plant tissue, plant disease suppression dramatically improves. Silicon supply to the plant must be continuous, or disease suppressive effects are reduced or non-existent. Silicon can influence many components of host plant resistance. That includes incubation period, number of lesions, lesion size. So silicon may augment susceptible and partial resistance almost to the same levels of complete resistance. And again, I come back to how long it takes in a breeding program to select certain traits. You may be able to incorporate one gene for resistance. It's hard to stack genes. And you get that one plant out there, it could still be attacked by other pathogens. So this would have utility. Silicon changes the response of the plant at both the physiological and molecular level. So it implies that there's an active role for silicon in more than one disease signaling pathway. It may suppress plant disease as effectively as fungicides and may be well suited for inclusion in IPM programs. But clearly there's more research is needed. Let me just say that I didn't do this alone. I've worked with a team of people from many countries and many locations to get at a lot of these answers. And some are in the audience right now. So,
well, thank you for your presentation.